Biotechnology, in the broadest sense, means changing living structures to produce something new. In a real sense, the most powerful biotechnology first arrived on planet Earth about 4 billion years ago. Today, the products of this first biotechnology can be seen everywhere, in the air, in the earth, and in the water. The story of how that technology has worked out over these 4 billion years is becoming well known on Earth. It is called evolution by natural selection. As the third millennium begins, scientists have learned some of the secrets of how nature manages this bioengineering. And today we are beginning to do some bioengineering of our own that is changing the Earth in surprising and potentially revolutionary ways. By far, the most important success of biotechnology on Earth is the existence of life itself in all its surprising variety. Earth's first biotechnology arose in the ancient ocean, where atoms like carbon and oxygen and hydrogen, sulfur and phosphorus and nitrogen bonded together to form molecules that stayed together long enough to replicate themselves over and over and over. At present, we are puzzled as to the details of just how that happened, but we are confident it did happen in ways we are now beginning to understand and even able to replicate. Now, one of the important things to notice is that nature's engineering, producing a cell, a tissue, and an organism that can stay alive and reproduce, is bioengineering at the atomic and molecular level, an atom, a molecule at a time. Scientists have discovered one of the most important keys to this atomic molecular engineering, and that key is the structure of a large molecule called deoxyribonucleic acid, DNA for short. DNA, biologists discovered a few decades ago, is shaped like a twisted ladder, a double helix. The rails of the ladder are sugar and phosphate groups, and the rungs are pairs of 4 common bases: guanine, adenine, cytosine, and thymine. And it is the sequence of these base pair rungs that are the codes to direct the assembly of a living thing. Any living thing and all living things, atom by atom, molecule by molecule. This code is how nature puts together bacteria, cells, tissues, wings, legs, hearts, eyes, brains, amoebas, oak trees, snakes, beetles, butterflies, giraffes, whales, chimpanzees, and human beings. When human beings first evolved on Earth some 3 million years ago, another new twist was added to bioengineering capabilities: the birth of intelligence, conscious thought, rational control. Using this new capability, humans managed to develop the first consciously programmed bioengineering feats. They discovered how to do things like select and modify seeds of wild plants for deliberate culturing in gardens and fields. And they learned how to tame wild animals and use their products like milk, meat, protection, and energy for human purposes. And the first agricultural societies were born. Now, this was bioengineering at the whole organism level. Humans got closer to the cellular level, though as yet they knew nothing of cells, when they learned how to ferment grains and fruits to make beer and wine, how to culture milk to make cheese and yogurt, and dry and salt meat for long storage. Over the centuries, they improved on all these early biotechnologies. They began to deliberately cultivate and breed new kinds of plants and new kinds of animals, living things never before known in nature. And they learned how some kinds of animals and plants were harmful to humans, and they invented ways to destroy them. Parasites like the pneumonia bacteria, the tuberculosis bacteria, poliovirus, smallpox virus, and many others. All of these early biotechnologies worked on a level of cells, tissues, and whole organisms, but with little detailed knowledge of how the technologies actually worked. Well, in the early 21st century, Such detailed knowledge is rapidly becoming available, and work on these levels is continuing and producing some dramatic breakthroughs. Cloning, for instance. Ordinarily, living organisms on Earth reproduce by sexual methods. The egg from a female combines with the sperm from a male, and the resulting fertilized egg develops into an embryo and then into an adult of the same species. The hereditary codes are carried in the egg and sperm in packets called genes. These genes are themselves sequences of base pairs on the DNA molecule. In normal reproduction, both the egg and sperm each contain only half the adult number of genes, but when the half number from the sperm combines with the half number from the egg, the resulting fertilized egg and the resulting baby and adult is thus a completely new and novel mixture of genes. Well, for centuries, humans have known how to clone plants by taking non-sexual cells and tissues from the roots or leaves or stems, culture them in soil or water, and grow new plants that are genetic duplicates of the parent plant. Clones, that is. Well, multicellular animals were harder to clone. Since almost all cells in an adult animal have the same complement of genes, just like plants, biologists long ago speculated that it might be possible to reproduce an animal from a cell other than the sperm or egg. Why couldn't you, for instance, take a skin cell from a mouse and use the information coded in its nucleus to produce a new mouse that would be exactly like the original mouse, or cow, or sheep, or a human? Well, in 1994, biologists in Scotland succeeded in cloning the first mammal, a sheep. Using special microscopes and micropipettes and probes, they took the nuclear material, which contained the DNA codes, from a mammary gland cell of an adult sheep. They carefully inserted this nuclear material into an egg cell of another sheep, from which the original nucleus had been removed. Well, after many failures, they found success, and the first cloned mammal, Dolly, was born. Since that time, over 500 other mammals, sheep, cows, goats, mice, have been cloned successfully. As yet, no human being has been cloned artificially, but that possibility is there, and it may be realized in the 21st century, even though many human governments around the world have already passed laws prohibiting such biotechnology. Getting closer to the atomic molecular level of bioengineering, humans are now experimenting with changing the genetic codes of plants and animals and bacteria. Nature has been doing this for 4 billion years. The results from nature's experiments are the present-day array of over 3 million separate species of plants and animals that inhabit the air, earth, and water, as well as the 10 to 20 millions of species that once lived on planet Earth but are now extinct. In laboratories like this one, human scientists are using machines like this newly invented gene gun to forcibly insert new genes into plant and animal embryos. When successful, the emerging organism will be different from its parents in particular ways that humans find helpful. For instance, genetic engineers have successfully inserted a special gene into soybeans that makes the plant resistant to specific herbicides. In the past, soybean farmers could not use the most effective herbicides to kill weeds in their field because the same herbicide that killed the weeds would harm the soybeans. Instead, the farmer would need to work the field 6-7 times a summer to keep the weeds at bay. Now, with a new genetically altered soybean, they can use the more effective herbicide only once or twice a season, greatly reducing their workload, saving fossil fuels, producing more soybeans per acre, delivering a superior product at the end of the season, and potentially saving more of the Earth's surface from agricultural encroachment. In other words, helping to preserve more wilderness areas and save more species from extinction. Over half the soybeans grown in the United States now use these genetically altered soybean seeds. There's a similar story with corn. A genetically engineered variety that needs less fertilizer and less herbicide to produce more corn per acre is now in wide use around the world. And one of the most important of the world's food grains, rice, is being developed with a special gene inserted to make vitamin A in the edible grain. This variety may turn out to be very important for consumers in some of the poorer countries on Earth, where blindness from lack of vitamin A is a serious problem. Work is also proceeding to add a gene to rice that would make it richer in iron. This would make the rice able to combat anemia, another widespread problem among women in poorer countries of the world. Just one more example. One of the world's greatest killers today is hepatitis B. It can be prevented, though, with a 3-shot course of inoculations that cost about $200. Well, most of the deaths from hepatitis B occur in poor countries where this cost is out of the question for most people. Well, researchers in the United States are having success now with inserting the genes that code for hepatitis B vaccine into a banana plant. If and when commercially successful, a life-saving inoculation for this disease, eating a banana, would cost less than 10 cents. In addition to genetically altering crop plants for more production, more nutrition, and for disease control, scientists are also inserting genes into plant species that can make them powerful pollution controllers. Plants are being genetically engineered, for instance, to make them able to soak up cadmium, copper, mercury, or arsenic compounds from the soil and bind them into their tissues. Then you harvest the plants and bury them where they can do no harm. Genetically engineered plants are also being developed that can produce industrial chemicals for a fraction of the usual factory cost and with much fewer and less difficult pollution problems. Biotechnologists around the world are combining gene engineering with cloning techniques. Cows, sheep, pigs, and mice, for instance, have all been genetically altered to produce particular hormones, enzymes, proteins, antibiotics, or other useful chemicals. These genetically altered animals, called transgenic animals now, are then cloned. Potentially, you have a whole herd of transgenic cows, for instance, that produce large quantities of human insulin in their milk. Or a whole farmyard of transgenic pigs that can provide whole organs able to be transplanted into humans. Or on a different level, the cellular level, a rich harvest of bioengineered human blood cells able to fight and defeat the AIDS virus. Or maybe even able to slow or stop the chemicals that cause Parkinson's or Alzheimer's disease. Some of the most important and most promising experiments in biotechnology on Earth use yeast cells or bacteria for their subject. Most people are not aware of the enormous populations of bacteria and the enormous number of different species of bacteria that far exceed the populations and species of multicellular plants and animals. In the human mouth alone, there are over 600 species of bacteria have been found. Well, some bacteria cause disease in plants and animals, but most bacteria perform essential jobs in cycling elements in the ecosystem of planet Earth. And bacteria are ideal subjects for the biotechnology lab. They're easy to engineer, easy to culture, fast to reproduce, and versatile in producing useful products. In biotechnology laboratories, scientists have learned to cut and paste genes into the DNA of bacteria, creating designer bacteria that can carry out specific tasks useful to humans. The common procedure is to insert a gene into the bacterial